Hi everyone, welcome back to another episode of Neurosci IQ. Today we are going to dive into the fascinating world of learning and memory formation. Specifically, we will be talking about long term potentiation, also known as LTP. If you ever wondered how our brains encode and retain information, or how neurons communicate to form long lasting memories, then you are in the right place. Essentially, what LTP is, it is a long-lasting increase in signal strength between two neurons. LTP is also heavily implicated in learning and memory formation, so it is very prevalent in the hippocampus tissue. As you can already see from the diagram of the big picture of the overview of LTP, there are many different changes that the synapse goes through as a result of long-term potentiation. Just to name a few, we can see there are more receptors, the receptors has changed conformations, as well as there are more neurotransmitters being released. There are two main types of receptors that we should be aware of when talking about long-term potentiation. The first of these receptors are the AMPA receptors, and the second one that we will get to in the next slide are the NMDA receptors. Essentially, AMPA receptors bind to glutamate, and as a result of binding to their neurotransmitter, they open up and they allow sodium to rush into the postsynaptic neuron and depolarize the cell membrane. So as you can imagine, the left image is just the sodium ions being stuck in the synapse, but as soon as glutamate binds to the AMPA receptors, the sodium ions can rush into the postsynaptic neuron and depolarize the cell. Now let's talk about NMDA channels. These channels are very similar to AMPA channels that we talked about, as they also bind to the glutamate neurotransmitter. However, there is one big difference. As you can see, under normal conditions, there is a magnesium ion that blocks these channels and prevents ions from passing through. Furthermore, NMDA receptors are both permeable to calcium ions and sodium ions. But after getting repeatedly stimulated, the synapse goes through changes and the magnesium ion is displaced. So you could say one of the changes that long-term potentiation causes is the displacement of the magnesium ions, which means that ions such as calcium and sodium can freely pass through the channels and enter the postsynaptic membrane. Now let's talk about the different pathways involved in long-term potentiation. Essentially, as we talked about, repeated stimulation of the AMPA receptors causes the magnesium ions blocking the NMDA channels to be displaced, which allows calcium ions to rush into the cell through the NMDA receptors, which depolarizes the postsynaptic neuron. And furthermore, LTP can be divided into two distinct phases. The first phase is the induction phase, which involves the mobilization of different proteins and enzymes, such as through phosphorylation. This phase typically happens within minutes of the initial event. However, the second phase is called the maintenance phase, and this phase involves the synthesis of new proteins and enzymes, as well as upregulation of genes. Therefore, this phase can take hours and happens after the induction phase. The other way to divide the LTP events is dividing the long-term potentiation into changes that happens to the presynaptic neuron, which releases the neurotransmitters, and also the postsynaptic neuron, which contains the AMPA and NMDA receptor and responds to the neurotransmitter release by the presynaptic neuron. And as we can see from the flowchart, there are different enzymes and different pathways that are involved, And in the next few slides, we will take a look and specifically focus on the events that happens in the postsynaptic neuron. One of the most important pathways for long-term potentiation involves protein kinase A or PKA. After calcium ions rush into the cell through the activated NMDA receptors, 4 of the calcium ions bind to calmodulin to form an activated calcium calmodulin complex, which activates PKA, and protein kinase A has a wide variety of functions. And just to name the few most important ones, one of the main ones is making new AMPA receptors. So there are actually more AMPA receptors on the cell membrane during LTP, which means that they are more ready to respond to neurotransmitter release by the presynaptic neuron. Furthermore, PKA allows the AMPA receptors to get phosphorylated, which means that they can remain open for a longer time even after glutamate is no longer binding to the AMPA receptors. So these changes are more long lasting because even if glutamate isn't there, the AMPA receptors that are phosphorylated remain open for a longer period of time. Furthermore, protein kinase A is responsible for inhibiting phosphatases inside the cell, which means that the AMPA receptors that were phosphorylated won't get dephosphorylated as quickly and they can remain open for a longer period of time. Another very important pathway for long-term potentiation involves the calcium calmodulin dependent protein kinase 2, and once again calcium calmodulin complex activates CAM kinase 2, and this leads to a wide variety of different downstream effects. One of the most important ones, of course, is the making of new AMPA receptors, which makes the postsynaptic membrane more sensitive to the neurotransmitter released in the future. But also, CAM kinase 2 is involved in upregulating genes for building new dendrites. And another very key aspect of CAM kinase 2 is actually the fact that it can autophosphorylate. And what I mean by this is even after calcium calmodulin complex is gone, and for example there is no calcium coming into the cells anymore because, for example, the receptors have closed, the autophosphorylation allows CAM kinase 2 to remain activated and continue having these downstream effects, which makes the LTP have long-lasting implications for the neuron. So far, we have mostly focused our discussion on the changes associated with the postsynaptic neuron. However, there are also changes to the presynaptic neuron that we should talk about. While this is more controversial and the exact mechanisms remain unknown, scientists think that the postsynaptic neuron can send a signal to the presynaptic neuron to tell it like, hey, we are going under long-term potentiation. You should also make some changes so next time we are even more stronger. And this signal that gets sent from the postsynaptic to the presynaptic neuron, it's called the retrograde signal because unlike most times, it is going from the postsynaptic to the presynaptic neuron rather than the presynaptic to the postsynaptic. And the retrograde signal could have many effects, but the most important ones that the scientists are curious about are the increased vesicle numbers, so more neurotransmitters are released and synthesized, but also there is also thought to be an increased probability to release the vesicles in the first place. Thank you for listening to this episode of Neurosci IQ. If you enjoyed this episode, please make sure to like and subscribe.